Welcome to Omni Academy. This is Maria, and today we are starting our third lecture on development of eye. In the last lecture, we studied the early morphogenesis of eye, and in today's lecture, we will be studying the development of lens. But before that, let me just revise the step of invagination because this step is important in the development of lens. The step of invagination converts the optic vesicle into a optic cup and lens placode into a lens vesicle. This lens vesicle is very important because this is playing the major role in the development of lens. As we have mentioned in the last lecture that lens is the derivative of lens vesicle, so without lens vesicle no lens is formed. So the major focus of our today's lecture will be the lens vesicle. So let's start our today's lecture. So let's just firstly talk about the source of lens development. The origin of the lens is the surface ectoderm because the lens vesicle is derived from the surface ectoderm. So now let's just start the process of development. So the first step in the development of lens is the formation of lens placode. You can see in this diagram that the optic vesicle is touching the surface ectoderm. This touching induces differentiation in the overlying surface ectoderm, and that differentiation converts the overlying surface ectoderm into a lens placode. So the formation of lens placode is the first step in the formation of lens. So now let's move to the second step, which is the formation of lens pit. You can see in this diagram that the lens placode is invaginating, and as it is invaginating, it is forming a pit known as lens pit. So this is the second step in the formation of lens. So now let's move to the third step. So the third step in the formation of lens is the formation of lens vesicle. So the lens vesicle is formed when the lens pit keeps on invaginating until it pinches off as lens vesicle. So you can see in this diagram that the lens placode is pinched off as lens vesicle because of the continuous invagination of the lens placode. Lens vesicle loses its contact with skin around 5th week. The lens vesicle is initially a hollow sphere of surface ectoderm. You can see in this diagram that it is just a hollow sphere and there is nothing inside it. So now let's move to the fourth step. By the 7th week, cells from the posterior wall elongate to form primary lens fiber. You can see in this diagram that the cells from the posterior wall are elongating and they are moving towards the anterior aspect, and these cells are known as primary lens fibers. The primary lens fibers fill the lumen and reach anterior wall by 7th week, thus obliterating the cavity. So this was the fourth step in the formation of lens. But let me just explain the whole process of formation of primary lens fiber. Cells of the posterior surface of the lens vesicle elongate until they completely fill the cavity of the vesicle. You can see this is lens vesicle. Cells of the posterior wall are elongating and they will keep on elongating until they fill the cavity of the vesicle. You can see in this diagram that the cavity of the vesicle is now filled and there is no longer any cavity in the vesicle. As this process of elongation is completed, these cells lose their internal organelles, including their nucleus, to form the primary lens fiber. So you can see in this diagram that these fibers don't contain any nucleus or any other internal organelle. This fetal lens becomes central part of the lens nucleus of the mature lens. It has the highest protein concentration and optical density. So you can see in this diagram that this area, lens nucleus, it is formed of primary lens fiber. So now let's move to the fifth step of the formation of lens. So the fifth step in the formation of lens is the formation of secondary lens fiber. Throughout life, these secondary lens fibers are added from the equatorial region. So let's now discuss the process of formation of secondary lens fiber. Cells around the periphery of the lens. You can see in this diagram that around the periphery of the lens, this area, there are anterior epithelial cells. These cells will undergo elongation, and after elongation, these cells will wrap around the lens nucleus. You can see in this diagram that the cells are undergoing mitosis, they are elongating, and they are now wrapping around the lens nucleus. You can see in this diagram. And as they wrap around the lens nucleus, they also lose their organelles and become the secondary lens fibers. You can also see the lens suture. This is the point where the fibers from the opposite sides of the lens meet. This process is largely complete by childhood but continues slowly throughout life. In cross section, these fibers are hexagonal and packed together somewhat like a honeycomb. So this was the fifth step in the formation of lens. Fibers in the middle are primary fibers and fibers in the periphery are the secondary fibers. You can see in this diagram of mature lens that the fiber in the middle are the primary fibers and the fibers in the periphery are the secondary fibers. Both the iris and the lens are covered by the iridopupillary membrane, but normally this membrane is resolved prior to birth. So let me just summarize the process of development of lens. So the first step in the formation of lens was the formation of lens placode. Second step was the formation of lens pit. Third step was the formation of lens vesicle. Fourth step was the formation of primary lens fiber, and the last or the fifth step was the formation of secondary lens fiber. So this was today's lecture. In the next lecture, we will be studying the development of iris. So see you in the next lecture.